One of the biggest subfields of astrobiology is alternative biochemistry, or how the fundamental chemistry of life that arose on another planet would compare and contrast with ours. There has been all kinds of hypothesizing about how different alien biochemistry could be from that of Terran life, from using molecules with slight modifications to the ones we do, to beings not even composed of molecular matter. In this video, we'll explore what I like to call the alternative biochemistry iceberg, with the biochemistries most similar to ours being at the tip, while the ones most different, and arguably the least feasible, are located at the base. But before we start, we should briefly go over Terran biochemistry. We use four main varieties of biomolecules, those being carbohydrates, proteins, lipids, and nucleic acids. Now, all of these are scaffolded around carbon, but also include other elements, namely oxygen and hydrogen, and this is why we're often referred to as carbon-based life. Starting with carbohydrates, the simplest form of these are sugars, with the ones being used by organisms having 5 or 6 carbons. These are often linked together or polymerized into larger chains. Carbohydrates are used for either energy storage, such as in the case of glycogen, or for structural purposes, such as in cellulose. Next, we have the proteins. These are long chains of smaller molecules known as amino acids. Each of these has a chiral carbon with a hydrogen atom, an amino group, a carboxyl group, and a variable R group bound to it. In proteins, there are a total of 20 different R groups, each of which defines a separate amino acid. The amino acids in proteins are linked amino group to carboxyl group. Proteins serve a variety of functions such as structural, signaling, or catalytic in the case of the enzymes. Of the 20 amino acids included in proteins, cysteine and methionine have sulfur atoms in them, and a few amino acids like ornithine and citrulline have roles in our biochemistry but aren't included in proteins. The simplest lipids are fatty acids, which have a carboxyl group linked to an alkane chain. Often, these are linked to a glycerol via an ester bond to form mono, di, and triglycerides. Sometimes, another functional group is attached to the glycerol, such as a phosphate group. This creates a polar head region in contrast to the hydrophobic alkane chain tail region, which in turn allows lipids to form bilayer membranes. Finally, we come to nucleic acids, which form our genetic material. These are composed of nucleotides linked together, and each nucleotide has a ribose sugar linked to a phosphate group on the 5th carbon and a nitrogenous base on the 1st. These nitrogenous bases are either adenine, guanine, cytosine, thymine, in the case of DNA, or uracil in the case of RNA. Apart from acting as the genetic material, RNA has been shown to have catalytic functions. To start down the alternative biochemistry iceberg, at the tip we have reverse chirality. When a carbon atom in an organic molecule is bound to 4 separate groups, it becomes non-superimposable over a mirror plane. This means that molecules with chirality have 2 stereoisomers, D and L. Different stereoisomers of the same molecules will have the same physical properties, but they refract polarized light differently. Organisms here on Earth use the L isomer of amino acids while using the D isomer of carbohydrates. It's been argued that life forms which rose independently on other planets could use the opposite stereoisomers to the ones we do. The abiotic synthesis methods we've developed produce an even mix of both the D and L isomers of these molecules, or a racemic mixture. However, the amino acids found in asteroids form a non-racemic mixture, with the L stereoisomer being slightly more common. It's not known for certain if this holds true in other solar systems, but it does suggest that if life originated elsewhere in the solar system, such as on Mars or Europa, it would also use the L stereoisomer of amino acids. One hypothesis for why astrogenic amino acids are non-racemic is the production of circularly polarized UV light in interstellar clouds. Whereas polarized light oscillates on a single plane, circularly polarized light oscillates on a rotating plane. When circularly polarized UV light strikes the precursors of amino acids such as carbon monoxide, carbon dioxide, ammonia, methanol, and water, it will produce amino acids of a specific stereoisomer. Indeed, experiments simulating interstellar conditions have shown that they give rise to non-racemic mixtures, where one stereoisomer can be 1.3% more common than the other. Specifically, circularly polarized light rotating clockwise gives rise to the D stereoisomer, while circularly polarized light rotating counterclockwise produces the L stereoisomer. On top of experimental evidence, we have observed non-racemic mixtures of propylene oxide in interstellar clouds. So, some regions of interstellar clouds would have greater concentrations of the D isomer or the L isomer, depending on which variety of circularly polarized light they are exposed to. As such, life forms which arose in different parts of the parent molecular cloud would be more inclined to use the D or L stereoisomer of amino acids in their proteins. But what about the chirality of sugars? Well, it has been shown that L amino acids can catalyze the production of D-glyceraldehyde from formaldehyde and glycoaldehyde. Early organisms could use this to produce the precursors of carbohydrates. So, it may be that the stereochemistry of carbohydrates and amino acids are intertwined, and that if an organism ends up using one stereoisomer of amino acids, it will use the other variety of carbohydrates, i.e., LL and DD amino acid carbohydrate combinations are unlikely. Now, let's go a little further down the iceberg to see what other varieties of the major groups of organic molecules, carbohydrates, amino acids, and nucleotides, organisms could use. Starting with sugars, or monosaccharides, there are 5 ones commonly used in biochemistry, that being glucose, galactose, fructose, ribose, and glyceraldehyde. But there are a wide variety of other sugars, both hexoses and pentoses, some of which are not used in any known organism, such as talose and idose. So, could these be used by alien organisms as an energy source or some kind of signaling purpose? Well, 2 other sugars, allose and altrose, have each been observed in one species of organism. The algae, Ochromis malhonemensis for allose, and the bacteria, Deutervibrio fibrosolvensis for altrose. How they produce these is not yet known, but they likely modified the aforementioned sugars which are common in Terran biochemistry. So, it's within the realm of possibility that simple sugars that are now only produced synthetically could be used by either Terran or extraterrestrial life forms. But what about a biochemistry that predominantly uses other sugars? If you boil formaldehyde, one of the simplest and most abundant organic molecules, it will result in the production of glucose and ribose. However, it will also produce many other simple sugars. So, was it that early organisms had access to a wide assortment of sugars and by chance chose to utilize glucose? Maybe, but maybe not. We've identified abiotic reaction networks which closely resemble glycolysis and the reverse gluconeogenesis, and the pentose phosphate pathway, the reductive version of which produces ribose. Under acidic and neutral conditions, the network much more closely resembles glycolysis, and under basic conditions, it's more like the pentose phosphate pathway. Ribose is required for the production of genetic material, meaning that so long as the molecular biology of an organism is RNA and DNA based, it may be that it will end up also utilizing glucose. But what about galactose and fructose? If I were to guess, I'd say that at an early point in the evolutionary history of life, around the time the last universal common ancestor lived, organisms evolved the ability to produce and metabolize these as well. And I can't really think of any reason why these should be favored over other unnatural sugars. So, while an alien's carbohydrate biochemistry would probably be based around glucose, monosaccharides, rare or non-existent in Terran biochemistry, could be widely utilized by them as well. But what about using other nitrogenous bases in their DNA? There are two varieties of nitrogenous bases used, purines and pyrimidines. Adenine and guanine are purines, and they base pair with their corresponding pyrimidines, that being thymine and cytosine respectively. However, adenine and guanine are one of many purines, most of which are used for other purposes in our biochemistry. These include caffeine and uric acid, which many animals use as a means of excreting nitrogenous waste. Furthermore, analysis of the Murchison meteorite has shown that along with adenine and guanine, many other purines are present. So, could organisms use these other purines instead? Well, one of the purines, hypoxanthine, is a precursor to both adenine and guanine. While the name of the purine is hypoxanthine, the nucleotide which contains it is inosine. Meanwhile, on the pyrimidine side, both thymine and cytosine are produced from uracil, which substitutes thiamine in RNA and has been found in asteroids. It has been shown that inosine can base pair with uracil, meaning one possibility is that alien organisms could have a much more quote-unquote simple molecular biology than ours, which only use these two. DNA can act as the genetic material because it specifies the amino acid sequence of proteins, and it does this with codons, a 3-nucleotide sequence which specifies a given amino acid in a protein. Why is it 3-nucleotide? It comes down to math. With 4 nitrogenous bases and 1 nucleotide, you could specify 4 amino acids. With 2, you could specify 16, and with 3, you could specify 64, and there are 20 amino acids used in proteins. This is why amino acids are often coded for by more than one codon. However, if there were 2 base pairs, a 3-codon sequence could code for 8 amino acids. With 4, it could specify 16, and with 5, it could code for 32. So, if an organism had a binary genetic code with only inosine and uracil, it would need at least 5 base pairs per codon to code for the 20 amino acids. This would mean it would need more genetic material to specify a given number of proteins. But, given that a lot of our DNA has no known function, this wouldn't have been much of a hindrance. If alien life did expand its genetic code, would it necessarily use adenine, guanine, thymine, and cysteine, or could it use similar molecules instead? Well, a modeling experiment has indicated that caffeine can substitute adenine in a DNA molecule, as it will more effectively hydrogen bond with thymine than cytosine. However, caffeine-thymine bonds are not as stable as adenine-thymine ones, or guanine-cysteine bonds. Furthermore, inosine can also pair with cytosine along with uracil, implying that if it was used in genetic material with more than one purine-pyrimidine pair, you'd often have mutations, with the wrong pyrimidine being substituted in the nascent strand during replication. This suggests that while it is possible that other nitrogenous bases could be used in an alien's genetic material, adenine, guanine, cytosine, and uracil, or thymine, were likely incorporated into our genetic code because their base pairing is most stable and extraterrestrial life would be more inclined to utilize them as well. Although, I should mention that another kind of binary genetic code could prove advantageous under certain circumstances. Adenine bonds to thymine with 2 hydrogen bonds, while guanine binds to cytosine with 3. As a result, the greater the GC content of a DNA helix, the tighter the 2 strands will be held together, and the greater the temperature needed to separate them, or the annealing temperature, will be. As a result, microbes which inhabit hydrothermal systems will usually have a higher GC content in their genome. However, there's only so much GC you can have before you can't use amino acids with adenine and uracil in their corresponding codons. However, if an organism lived on a planet where the surface temperatures were always in excess of 75 degrees, perhaps they'll avoid this problem by having a 5-nucleotide codon GC binary genetic code. But whatever kind of code an alien's biochemistry uses, would it also code for the 20 amino acids we use? Or would it incorporate others into its proteins? One argument for them using the same 20 amino acids is that even though amino acids like the aforementioned ornithine and citrulline are present in our cells, they aren't included in proteins, and if it was advantageous to do so, compared to the 20 proteogenic amino acids, they would be included in the proteins. Although, citrulline is an interesting case, because while it isn't incorporated into a peptide during translation, post-translation, sometimes arginine residues are converted into citrulline, and it is thus a quasi-proteogenic amino acid. It's a similar story with isoserine, which, while not included in the genetic code, can be part of a protein via the modification of a serine residue. So, I wouldn't find it odd if an alien organism simply included citrulline and isoserine in its genetic code. Furthermore, I said that there were 20 amino acids included in proteins during translation, but this term isn't entirely accurate. There are 20 amino acids that are universally used by all organisms, but there are 2 more, selenocysteine and pyrrolysine, which are used only by some organisms. Also, as many as 118 non-biogenic amino acids have been included in engineered proteins. All this to say, it's at least theoretically possible for alien life to include other amino acids in their proteins. But what other amino acids would they have had access to? In the famous Miller-Urey experiment, it was shown that passing electricity through a mixture of reduced gases, simulating lightning in what was thought to be Earth's primordial atmosphere, it would produce some, but nowhere near all, of the amino acids we use. Further experimentation with a similar design has been able to produce more of the amino acids included in proteins, along with many other non-proteogenic ones, but not all of the amino acids which are coded for, namely arginine, lysine, and histidine, were observed, implying that these were first synthesized biotically and later incorporated into the genetic code. So, of the amino acids that could have been produced on the primordial Earth, did early life just randomly choose which ones to include in its ancestral genetic code and later expand it with newly biosynthesized ones? Again, maybe not. While we nominally think of the TCA cycle as a means of producing energy, many of the intermediates are alpha-keto acids, and these are converted into amino acids via transamination reactions. More specifically, pyruvate is converted into alanine, alpha-ketoglutarate is converted into glutamate, and oxaloacetate is converted into aspartate. These amino acids are then modified into similar ones like cysteine, glutamine, and asparagine. In a manner similar to glycolysis and the pentose phosphate pathway, we've observed abiotic analogues of the TCA cycle, and unlike the Miller-Urey experiments, these occur under biologically relevant conditions. This means the intermediates could have been produced by early cells. So, it may be that organisms would be inclined to use the amino acids which can be immediately produced from TCA cycle intermediates. Since many other amino acids are produced from these via varying degrees of modification, the proteogenic amino acids most similar to the TCA cycle intermediates would be most likely to be used by extraterrestrial life. Conversely, the less they resemble these, the less likely it is to be used, and instead an alien organism would use an amino acid which its ancestors produced de novo. Now, on to the last group of biomolecules, lipids. Simple carboxylic acids can form bilayer membranes, and are easy to produce in prebiotic conditions. Furthermore, imidazole and cyanamide will catalyze the synthesis of mono, bi, and triglycerides, and when these are present along with ammonium and phosphoric acid, it will result in the production of phospholipids. Even though carboxylic acids can form membrane-bound vesicles, they are too permeable and unstable to form the membranes of modern cells. So, I'd argue that it's likely that organisms would use phospholipids centered around a glycerol linked to alkane chains via ester bonds, initially at least. But could they wind up using other kinds of lipid membranes down the road? To answer that, we need only look at prokaryotic organisms. Bacteria often have two membranes in their cellular envelope, with the inner one being composed of phospholipids similar to the ones we use. Meanwhile, the inner layer of the outer membrane is composed of phospholipids, but the outer layer is largely made of lipopolysaccharides. These are composed of two sugars linked to carboxylic acids by either ester or peptide bonds. The lipid membranes of the other major group of prokaryotes, archaea, are even more bizarre. These are also built around a glycerol, but instead of being linked to alkane chains via ester bonds, they're bound to polymerized isoprenoids by ether bonds. This gives the hydrophobic region branches coming off the main chain, and these form a monolayer membrane with a hydrophilic region at each end of the molecules. This discrepancy has led to a lot of speculation as to what kind of membrane the last universal common ancestor had, with all sorts of odd combinations of bacterial and archaeal lipids being proposed. However, this kind of discourse ignores some very basic principles of evolutionary biology. Most notably, there's no rule which states the ancestral character state has to be an even mix of that of the descendants. One member of a clade can more closely resemble the common ancestor than the other, and given that bacterial-type phospholipids are easier to synthesize abiotically, I'd argue that Luca had a similar membrane, with the archaeal-type membrane evolving later. However, if the bacterial branch died off shortly after Luca, with all organisms being archaea, all the lipids used in membranes would be the isoprenoid ether-linked ones. So, while it's pretty likely that alien organisms would end up utilizing phospholipid membranes with alkane chains and ester bonds independently, we know it's possible for them to wind up using other kinds of lipids in their membrane. As such, we'd probably find phospholipids similar to ours when, or if, we discover alien biospheres, but some would probably have other kinds of membrane lipids unique to them. One thing to note about the separate varieties of membrane lipids used by Terran life is that they all have a phosphate group at their head. So, it's pretty safe to assume that whatever kind of membrane lipid an alien organism uses, it would have a phosphate group as well. Or is it? Phosphorus is one of the major biophilic elements, and it fills a variety of roles in our cells. Of course, there is the phosphate group in the membrane lipids, as well as being incorporated into the backbone of genetic material, and it's deeply intertwined in our energy metabolism. ATP is often described as the universal energy currency of life, and it is able to fill this role because the phosphoanhydride bond between the second and third phosphorus atoms is readily cleaved, resulting in the phosphorylation of organic molecules, and thus making them more reactive. But many astrobiologists have proposed that these roles could instead be filled by arsenic. When two elements occupy the same column on the periodic table, it's because they have electrons in the same arrangement in their outer orbitals, i.e., they have similar chemical properties, and arsenic is in the same column as phosphorus. Now we come to the waterline of the alternate biochemistry iceberg, and see how suitable arsenic really is as a substitute for phosphorus. Modeling experiments have shown that if you swap phosphorus out with arsenic in DNA, it will exhibit similar conformational properties, and, while there will be some changes to the internal geometry of the strand, i.e. angles and distances between the constituent atoms, it wasn't sufficient to alter the distance between the nitrogenous bases. Thus, base stacking is maintained, and the DNA molecule should be able to fulfill its original purpose, theoretically at least. Although, the greater reactivity of the arseno-diester bond would cause the strand to be more unstable. As such, it may be that arsenic-linked genetic material is simply too unstable for life to utilize. Or, at least life is simple as the simplest organism alive today. But remember, in the distant past, there were even simpler organisms. So, perhaps very simple protocell-like organisms could be arsenic-based. But, if it's more reactive than phosphorus, wouldn't it make for a better component of the energy currency than phosphate? Well, finding the optimal energy currency via natural selection is a bit of a balancing act. If a functional group is not reactive enough, it obviously can't be used to drive an organism's metabolism, the metabolism being the sum of all reactions occurring within it. Conversely, if it's too reactive, one of two things can happen. First, it will more readily react with molecules that aren't targeted for activation, essentially chewing up organic matter. And second, it will more rapidly spontaneously hydrolyze, basically wasting energy. It's likely that the phosphoanhydride bond in the ATP molecule represents an optimal balance between these two extremes, while an energy currency with an arsenic-anhydride bond would just be too reactive. You could argue that such an energy currency would be viable at cooler temperatures, but the problem with this is that pretty much everything we know about prebiotic chemistry suggests that it requires temperatures greater than ambient atmospheric temperatures, but less than the boiling temperature, meaning it's doubtful that such a biochemistry could arise in the first place. As for being part of the polar heads of lipid membranes, many organisms on Earth are known to use arsenolipids, or lipids with an alkane chain and an arsenate head group. However, these are believed to be involved in arsenic detoxification and don't form membranes, and as far as I know, no one has been able to produce an arsenolipid bilayer membrane in a laboratory setting. One problem with using arsenic instead of phosphorus has to do with its relative abundance, with phosphorus being more common in both the universe and Earth's crust. Although, there are localized environments where it's present in greater concentrations. Certain environments like the saline Mono Lake in California have high concentrations of arsenic, and one strain of bacteria isolated from it, known as GFAJ1, was subjected to an experiment where it was either provided arsenic and phosphorus, arsenic but no phosphorus, phosphorus but no arsenic, or neither arsenic nor phosphorus. In the arsenic and no phosphorus treatment, cell growth was observed for 4 divisions, while only 1 division was observed with neither arsenic nor phosphorus. This seemingly indicated that the bacteria were using arsenic to perform the roles phosphorus does in other organisms, and this was corroborated by nanosims, which showed the DNA had arsenic in it. However, shortly after the results of this experiment were published, it quickly came under scrutiny. One criticism is that the arsenic provided could have been contaminated by phosphorus, which would have facilitated cell growth. Furthermore, the DNA wasn't washed with ethanol to remove other molecules before being subjected to nanosims, so the detection of arsenic could have been an artifact. Although, analysis of the lipid composition showed unusually high concentrations of arsenic in the cells not provided with phosphorus, many of the critics of the publication claim that because the notion that the cells were incorporating arsenic into their DNA is not sustainable, the idea of arsenic-based life is dead in the water. They go on to say that the ability to grow in the arsenic-only media was largely the result of swapping out some of their phospholipids with arsenolipids, freeing up more phosphorus to be used in the genetic material. But even if that is the case, the cells are still swapping one function of phosphorus out with arsenic, and thus give some credence to the possibility of alien life using arsenic for some functions Terran life uses phosphorus for. Although we have no clear answers at this point, which is why I like to call this the waterline of the alt-biochem iceberg. Above this point, we can use comparative biochemistry to get an idea of how extraterrestrial biochemistries would function, while below it, it's pretty much all speculation. However, with arsenic-based biochemistry, the data we have available is inconclusive, hence it's the dividing line between these two regions. Just below the waterline, we have alternate kinds of energy currencies. while extraterrestrial life will almost certainly use another kind of molecule besides ATP. It's been proposed that instead of the phosphoanhydride bond and resulting organophosphate, a different kind of functional group could be used. One of the most frequently discussed alternatives is a thioester group. Sulfur is more abundant in the universe, and while it's not more abundant in the crust of the modern Earth than phosphorus, it would have been more common in the hydrothermal systems where it's believed that life begun. The main reason that energy metabolism is largely centered around phosphorus is because phosphates are such a good leaving group. Essentially, the oxygen between a carbon atom and the phosphorus will hog the electrons, opening up the carbon for a nucleophilic attack by a functional group of another organic molecule. This is why adding an organophosphate group makes organic molecules more reactive, along with readily facilitating dehydration polymerization reactions. Because the phosphoanhydride bond in an ATP molecule is even more unstable or high energy, it can be used to phosphorylate organic molecules. However, instead of using a phosphate ester bond, it may be that some organisms instead activate organic molecules by reacting a thiol bond with a carboxylic acid to form a thioester. Thioesters are a better leaving group than alcohols, so they could theoretically serve as an energy currency, but they aren't as good as organophosphates. As such, any organism who used them would probably have a lower metabolic rate than those on Earth, at least assuming that they came about in a similar chemical environment. As stated before, finding the best energy currency is a balancing act, but chemical conditions can alter reactivity. For example, higher temperatures and pressures coupled with lower pHs will accelerate hydrolysis reactions. So, if a planet had a higher surface pressure, it would result in the surface water having a higher boiling point, with hydrothermal systems having temperatures in excess of 100 degrees Celsius. If life begun in an acidic hydrothermal system on such a planet, organophosphates may prove too reactive in these conditions, so instead of using a phosphoanhydride bond as its energy currency, it would be advantageous for life here to use thioesters. While we've discussed the possibility of life using other nitrogenous bases in its DNA, another possibility is that life wouldn't use DNA at all. There are alternative xenobiotic nucleic acids, or XNAs, which are often hypothesized to use a different sugar instead of ribose. Some of these include locked nucleic acids, LNAs, hexatyl nucleic acids, HNAs, threose nucleic acids, TNAs, arabinonucleic acids, ANAs, cyclohexanol nucleic acids, CENAs, 2-fluoroarabinonucleic acids, FANAs, and homodNA. All of these will base pair with a single-stranded DNA segment except homodNA, because its molecular geometry is more similar to a ladder versus a helix, although most XNAs can't complementary base pair with a strand of DNA. It has been shown that both RNA, nucleotides, and polymers could be produced abiotically on the early Earth. As such, there's an argument to be made that life would probably end up using RNA at first, but later utilize another kind of genetic material. And I guess you could say that DNA is a different genetic material, but it only has a minor modification, with the hydroxyl group on the second carbon being replaced with a hydrogen. In fact, it could be said that due to the instability of RNA, it could have been an inevitability that it will be replaced by something more stable, and due to their chemical similarities, DNA would be the best candidate. But if it is the case that life uses RNA at first, any number of XNAs could replace it as the genetic material, so long as it could complementarily base pair with it, ensuring that the genetic information is passed from the RNA strands to whatever XNA is used. The RNA world hypothesis postulates that when life begun, RNA acted as both genetic material, what DNA does, and a biocatalyst, what enzymes do now. However, a major problem with RNA acting as the genetic material is the aforementioned instability, which may be too great for even a protocell-like organism to use it in its genome. As such, it's been proposed that another variety of nucleic acid was used as the genetic material, which was transcribed into RNA, which instead performed the function enzymes do today. Both tRNA and peptide nucleic acid, or PNA, can be produced abiotically under early Earth conditions and are more stable than RNA. So, it may be that the first cells used one of these as its genetic material, which was then transcribed into an RNA sequence, with a resulting catalytic RNA sequence, or ribozyme, facilitating early metabolic functions. Later, the genetic information could be transcribed into DNA, which replaced either PNA or tRNA as the genetic material. And this brings me to a point that's often lost in this discussion. Even to this day, organisms do use RNA for biocatalysis. Most notably in the assembly of peptide sequences making up a large portion of the ribosomes. This raises an obvious question. Could XNAs have catalytic properties as well, which could be utilized by alien organisms? We've recently synthesized DNA sequences which had catalytic function, so we know RNA isn't alone in this regard. As far as I know, catalytic functions of XNAs has not been observed, but given that ribozymes catalyze biotic reactions via interactions between the nitrogenous bases and the substrate, it should be possible for other varieties of nucleic acids to perform similar functions. Apart from using a different sugar, it's been hypothesized that life could use a substitute for the phosphodiester group, which links them. We've already discussed arsenic, but other alternatives include sulfone, PS, NP, DFO, and triazole groups. Sulfone DNA can form short chains, or oligomers, but if they get too long, self-aggregation occurs. Substitution of the nitrogenous bases can make this worse, indicating that sulfone couldn't act as a substitute for a phosphodiester. However, long, stable strands of DNA have been produced with the phosphodiester group swapped out by a phosphothionate, or an N3P5 phosphoamidate, suggesting they could be used as a backbone linker. A similar molecule to NP-DNA, known as P3N5 phosphoramidate DNA, can form stable polymers, but it's more stable than NP-DNA under basic conditions, while being less stable under acidic ones. So, if alien life was to use one or the other, which one it utilized would depend on whether it arose in a basic or acidic environment. Two other nucleic acids which can form stable strands are the triazole and DFONAs. While carbon acts as the main scaffolding agent for biomolecules, many other elements constitute them, with organic matter having roughly 12 hydrogen, 6 oxygen, and 1 nitrogen atom for every 6 carbon atoms. One proposed alternate biochemistry is hypothesized to have boron as another major biophilic element, with relative abundances between that of oxygen and nitrogen in Terran life. Organoboron chemistry is a major subfield in organic chemistry, but boronated compounds are very unstable under oxidizing conditions. However, they are far more stable in reducing ones. So, such a biochemistry would be more suitable on a planet with a hydrogen atmosphere. Although not a major player in our biochemistry, some biomolecules have boron in them. These include quorum-sensing molecules employed by some bacteria and cross-linking molecules in plant cell walls. Furthermore, borate is shown to catalyze the production of ribose from glycoaldehyde and formaldehyde, nucleotides from ribose, urea, and phosphate, and peptides from amino acids. But once the first protocells begun to form and evolve, boron may have just been too reactive to play a major role in their biochemistry. So, it could be that had this happened in an environment where boron had been more stable, life would have included it in many functional groups within its version of enzymes to facilitate metabolic reactions. Let's shift gears a bit. So far, we've talked about how the molecules life is made of could be different versus the media it's in. But, apart from being carbon-based, life on Earth is said to be water-based, and that biochemical reactions occur while they are dissolved in water. Yet, water is one of many liquids which are fairly common in the universe, and some of these could also serve as liquid media for biochemical reactions. The most frequently discussed alternative is ammonia, which is also polar and will thus interact with organic molecules in similar ways to water. Furthermore, a number of reactions involving organic molecules have been observed in liquid ammonia. Some of the best studied include nucleophilic substitution and metal ion catalyzed reactions, both of which are staples of our own biochemistry. Also, organic acids can be deprotonated by the ammonia molecules, increasing ionization rates. However, rates of nucleophilic substitution in ammonia are lower than they are in water, at least for aromatic groups. But getting organic molecules to react in a solvent is one thing, and getting them to constitute a metabolic network is another. Ammonia boils at negative 33.34 degrees Celsius, and, as stated earlier, much of what we know about prebiotic chemistry indicate temperatures between 50 and 100 degrees Celsius are the most conducive for life to begin. When we look at the optimal growth rates of prokaryotes adapted to different temperatures, the greatest growth rates are observed in the thermophiles at around 67 degrees Celsius. Both of these observations indicate that there may be an optimal temperature to kickstart metabolism, roughly between 50 and 80 degrees Celsius. Below this temperature, prebiotic molecules don't react fast enough, and above it, they can't form the molecular structures characteristic of life. So, what does this mean for the possibility of ammonia-based life? Well, it means life is unlikely to begin in a world with bodies of liquid ammonia that has a similar surface pressure to the Earth, because the range of temperatures it exists as a liquid are probably too low to kickstart metabolism. But, like water, the boiling point of ammonia increases with pressure. At a surface pressure of 11.35 megapascals, or 112 atmospheres, the boiling point of ammonia is 132 degrees Celsius. If you increase the pressure and temperature above this point, it becomes a supercritical fluid. At a pressure of 62 megapascals, ammonia has the same boiling point that water does here on Earth at sea level. At 40 megapascals, it has the boiling point of 77 degrees, which would be enough to support prebiotic chemistry. So, it may be that in order for ammonia-based life to start, it would have to be on a planet with an atmosphere thick enough to keep ammonia liquid at the required temperatures. Another possibility is that a planet could have a somewhat greater surface pressure, say around 10 megapascals, with ammonia oceans, and while the temperatures near the surface, say less than 10 degrees, are too cold for life to begin, deep down the pressures and thus the boiling point of ammonia increases. Here, deep-sea hydrothermal emissions would heat the ammonia sufficiently for prebiotic chemistry to begin. Life there would initially propagate among similar hydrothermal systems until it developed adaptations to be able to carry out metabolism at lower temperatures, similar to the psychrophiles on Earth. At this point, it could colonize the surface world. However, one problem with this scenario is that what we know about prebiotic chemistry indicates that wet-dry cycles are a key driver of it, and this wouldn't happen in a deep-sea environment. But then again, it could be quite different for ammonia-based prebiotic chemistry. Now, this raises an interesting question. How would biomolecules used by ammonia-based organisms differ from those employed by water-based life? First off, I'd argue that the positions nitrogen and oxygen occupy in the elemental stoichiometry of life would be flipped, with nitrogen being almost as common as carbon itself. These would likely be in the form of amino or nitrile groups. In terms of forming cellular structures, life on Earth depends largely on how water interacts with its constituent molecules. For example, enzymes will fold the way they do because water is attracted to the hydrophilic R groups on the residues that end up on the surface, while repelling the hydrophobic ones that go into the interior. And of course there's the formation of the lipid bilayer membranes. Since ammonia isn't as polar as water, it may be difficult for an alien's version of proteins or membranes to aggregate in a given way, so they may have to use groups with charged atoms on the side facing the solution and a larger nonpolar region. At larger scales, an ammonia solvent for life would have other challenges as well. For one, ammonia has a lower heat capacity, requiring 2.16 kilojoules to heat 1 kilo of ammonia 1 degree Celsius, compared to water's 4.18. On top of that, once you get it to the boiling temperature, it requires 2,260 kilojoules to transition 1 kilo of water from liquid to gas, while it takes 1,371 kilojoules for the same mass of ammonia. As such, large bodies of water will have much steadier temperatures and be more resilient to evaporation than large bodies of liquid ammonia. Apart from ammonia, other solvents have been proposed. One of these is hydrogen sulfide. This one may seem like a good candidate for life to use, given its geometric similarities to water, with sulfur being the same column in the periodic table as oxygen. Like ammonia, it's a gas at room temperature, with a boiling point at negative 60 degrees Celsius, but it can exist as a liquid at temperature ranges similar to water with higher pressures. The greatest temperature it can exist as a liquid at is about 100 degrees Celsius at a pressure of 8.5 megapascals, or 84 atmospheres. Hydrogen sulfide is also weakly polar, even when compared to ammonia, meaning the aforementioned issues faced by ammonia-based life would be even worse for hydrogen sulfide-based life. On top of this, hydrogen sulfide is a poor solvent for inorganic materials, and metal ions are key catalysts in biochemistry. Just as ammonia-based life would probably have more nitrogen-based functional groups than water-based organisms, hydrogen sulfide-based life would probably have more sulfur-based ones. One of the biggest hurdles to either ammonia or hydrogen sulfide-based life is the relative abundance of nitrogen and sulfur when compared to oxygen. Even though they're both reasonably common, oxygen is the third most abundant element in the universe after hydrogen and helium. So, while planets with large quantities of ammonia and hydrogen sulfide on their surface will be common, it's hard to get a situation where you have an abundance of those with no water. As a result, oceans made up of either of these compounds are unlikely, and instead they'd be mixed with water. The ices which cover the surface of many bodies in the outer solar system appear to be mixed water and ammonia. Such a mixture would likely be too acidic in the case of hydrogen sulfide, or too basic in the case of ammonia, for pre-metabolic reactions to happen. Another proposed solvent is hydrogen fluoride, but fluorine is arguably too reactive to fill such a role and would end up chewing up the biomolecules. On top of that, it's pretty rare in the universe and is thus unlikely to form liquid bodies. All of the solvents discussed so far are polar, but some astrobiologists have argued life could employ nonpolar solvents as well. The most widely discussed one of these is methane, which is again a gas at room temperature, but unlike ammonia and hydrogen sulfide, high pressures can't make it liquid under biologically relevant temperatures, with its critical point being at negative 83 degrees Celsius. So, if methane-based life is possible, it would mean many assumptions we had about what the temperatures needed to kickstart life were mistaken. However, methane is one of many hydrocarbons, with some, like hexane and octane, being liquid at temperatures and pressures which occur on the Earth's surface. We know the formation of liquid hydrocarbon surface bodies is possible because many such bodies exist on Titan, which are composed of methane and ethane. Because the surface temperature of Titan is negative 183 degrees Celsius, it's probably too cold for life to begin there. However, there are a hypothetical variety of planets known as carbon planets, which would have large liquid bodies composed of hydrocarbons like hexane and octane. Since these are nonpolar, we don't have to worry about them mixing with any water that may be present on the planetary surface. Such planets would form in a protoplanetary disk with carbon to oxygen ratios above 0.65, and a recent study indicated that 12% of Sun-like stars would have formed in such a disk, implying carbon planets are fairly common. But what would biochemistry in a hydrocarbon solvent be like? For one, carbon, along with hydrogen, would probably be more abundant, while the relative abundances of oxygen and sulfur would likely be reversed, due to the greater solubility of hydrogen sulfide in nonpolar liquids when compared to water. The arrangement of molecular structures like enzymes and membranes would probably be reversed, with a polar region on the inside and nonpolar region on the outside. One problem that might arise with prebiotic chemistry in a hydrocarbon solvent is the insolubility of metal ions. These play key roles in biocatalysis even to this day. However, a variety of organic molecules known as vitamins are often at the active site of an enzyme where they aid in catalysis. The two main varieties of vitamins are the water-soluble ones, that being vitamin C and the B vitamins, and the fat-soluble ones, A, D, E, and K. We've identified methods by which B vitamins can be produced abiotically on the early Earth, so it may be that in a hydrocarbon liquid body, fat-soluble molecules with catalytic functions could form and facilitate prebiotic reactions in lieu of metal ions. Going back to the molecules life is made of themselves, we come to the most widely discussed variety of alternate biochemistry, that being silicon-based life. In a manner similar to phosphorus and arsenic, carbon and silicon are both in the same column in the periodic table. As such, it's been proposed that silicon could act as the main scaffolding agent for biomolecules instead of carbon. But, while carbon and silicon may seem similar at first, being able to bond with 4 other atoms, silicon's coordinate numbers are 3, 4, 5, and 6, while carbon's are 1, 2, 3, and 4. This results in different bond geometries, meaning that you can simply swap out carbon with silicon in biomolecules and expect it to act in a similar way, and more likely than not, it will break apart. For this reason, carbon and silicon can be thought of as false twins. Since it is tetravalent, silicon could in theory form a variety of molecules with a structural diversity on par with carbon, but these would be very different from our biomolecules. Furthermore, silicon-based macromolecules can have many functional groups, including silicon oxygen, silicon carbon, silicon nitrogen, silicon sulfur, and silicon phosphorus. Although double bonds between silicon and any other atom are very hard to form and unstable. Double bonds are very common in biomolecules, such as in carbonyl and aromatic groups. So, silica-based macromolecules couldn't have the same functional diversity as carbon-based ones. But while such macromolecules are possible, being able to exist in theory, or a vacuum, and in a solvent are two different things. Silicon is less electronegative than carbon, which means when it's bonded to anything else but oxygen while immersed in water, the hydroxyl ions dissolved in it will quickly perform a nucleophilic attack. This results in the formation of silicic acid and silicon dioxide. So, any silicon-based organisms wouldn't use water as their solvent. A proposed workaround for this is to use a low-temperature aprotic solvent like diatomic nitrogen or methane. Intuition would state that the lower temperatures couldn't kickstart pre-metabolic reactions. However, finding the optimal conditions for the origin of life is a balancing act, because if the system isn't reactive enough, it can kickstart prebiotic chemistry, but if it's too reactive, macromolecules will be too rapidly broken down. Because silicon-based macromolecules are more reactive than carbon-based ones, conditions in cryosolvents would tone down this reactivity, putting them in the optimal range for prebiotic chemistry. However, one problem with this scenario is the low solubility of pretty much anything in cryosolvents. For example, even the shortest silanes, Silicon-based analogues of alkanes, such as silane and disilane, only have a solubility of a few parts per thousand in liquid nitrogen, while organic molecules would require concentrations in the molar ranges for biochemistry to function. As such, both the reactivity and insolubility factors render silicon-based life chemically impossible, even though it could in theory produce large complex macromolecules. But it may not be completely game over for the inclusion of silicon in biochemistry. Some biomolecules have been artificially modified to have the carbon atoms swapped out with silicon, which are stable under biologically relevant conditions. So, it may be that an alien biochemistry could include silicon as part of an overall carbon-based one. It probably would be about as common as sulfur and phosphorus is in our biomolecules versus nitrogen and oxygen. Furthermore, it has been shown that while they can't form macromolecular structures of a size and complexity on par with proteins or nucleic acids, in a sulfuric acid solvent, silicon-based molecules are more stable than in water, and they can have a greater diversity of functional groups. Sulfuric acid has been seen as an implausible solvent for life because it too rapidly destroys biomolecules. However, in sulfuric acid, electrophilic attacks dominate, and the electron-poor nature of silicon, which makes it susceptible to nucleophilic attacks in water, makes it largely immune from this. Even though life couldn't be based on silicon alone, a silicon-carbon hybrid biochemistry could be viable, with carbon adding the necessary functionality and flexibility, and silicon adding structure and stability to the biomolecules in a sulfuric acid solvent. This is one way life could possibly exist in the Venusian clouds, which are composed mostly of sulfuric acid. Also, instead of being silicon-based, the xenomorphs could have this hybrid biochemistry, and it would explain why their blood is acidic. Another proposed scaffolding agent for biomolecules is sulfur, but this is unlikely because while sulfur can bond with as many as 6 different atoms in cases like sulfur hexachloride and sulfur hexafluoride, if it's bonded to more than 2 atoms, the extra atoms can only be bound to the sulfur atom. As a result, sulfur-based molecules can only form chains versus the complex 3D structures characteristic of biomolecules. Even more bizarre is heteropolymetallates biochemistry. Heteropolymetallates are metal ion oxides which contain many varieties of metals in various redox states. These can form a wide variety of crystal structures and can act as catalysts. So, it's been proposed that large crystals of a specified composition could act as enzymes, with other crystal structures forming the alien's version of membranes and genetic material. Such beings would likely have a crystalline shape, something akin to how hypothetical silicon-based organisms are often depicted. But there's no evidence that these could interact with each other in a way that would allow them to act like biomolecules. Now, we come to the bottom of the alt biochem iceberg with some truly esoteric ideas with life composed of non-molecular matter. One of these is plasma crystals which can form helical vortices. In a cold plasma medium, dust particles will crystallize, forming what are known as plasma crystals. The simplest one of these are spherical in shape and begin as a homogeneous cloud of dust grains. But over time, they develop a sort of structure as the particles slow down, transferring their energy to the surrounding dust particles. This structure has the fastest moving particles accelerated by the Langevin force on the outside of the sphere, while the slower ones remain on the inside due to the increased friction. As more crystals form and absorb dust grains, they become isolated from each other by a void without grains. However, spherical structures will only be stable if the external conditions are radially symmetrical, which they usually aren't. Otherwise, grain pairing will occur in the crystals, forming cylindrical structures, and, should they become stretched out, they will more closely resemble a helix with fewer grains per unit length. Sometimes, a slight change in the diameter of the helix induced by an external force will induce a change in the helical winding. This results in a bifurcation or heterogeneity in the structure of the helix, where two regions have different equilibrium diameters and winding angles. When dust grains are around the helices, they often form vortices around the bifurcation, and when it's near another helix, the vortex can induce a bifurcation in the second helix, thus giving it the same topology as the first. In this sense, the helices are replicating themselves. Such entities are hypothesized to exist in the nebulae, which cover vast tracts of interstellar space. Perhaps the best biological analog to this would be prions, or pathogenic proteins that are misfolded, which spread by inducing the same misfolding in other proteins that they come into contact with. From there, we come to the very bottom tip of the iceberg, with organisms not composed of matter, but magnetic poles and cosmic strings which inhabit stars. Magnetic fields will have north and south poles, which can be linked together via cosmic strings, resembling beads on a necklace or atoms in a linear molecule. Because they must have a specified sequence, alternating north-south, north-south, these strings couldn't convey any information unique to them, and thus could not be considered lifelike in any way. Furthermore, the poles will be attracted to each other, and thus end up self-annihilating. If magnetic monopoles existed, then the string could have a non-alternating sequence, and thus each string would be unique, but magnetic monopoles are impossible. But one possibility is the formation of magnetic semipoles, which don't have a net zero charge and can repel versus attract each other, preventing self-annihilation. With two possible semipole arrangements, which we'll call northwest and southeast, one of the two in the pair can be in a string, giving it four possible semipoles for a given position. It's something akin to our DNA, which can have one of four nitrogenous bases in a given position. The northwest and southeast pairing would also cause the corresponding semipole to align with it when it becomes linked to another chain, in a manner again reminiscent of our DNA, with adenine-thymine and guanine-cytosine pairing. Perturbations can then separate the resulting ladder structure, and thus the information in the first string is preserved in the second. In other words, it's replicated. Theoretical calculations indicate that this would occur in the stars, where the swirling plasma results in the formation of magnetic poles and semipoles. Also, the atoms in the plasma could catalyze the formation of beads. It's been shown that montmorillonite clay can catalyze the production of RNA polymers. So, the stellar plasma could be seen as an analog of the montmorillonite clay, and the processes described would be comparable to the late pre-biotic stage of biochemistry. The sequences which would replicate the fastest would outcompete the others for the building material, that being magnetic semipoles, thus giving rise to Darwinian evolution. Getting even more theoretical, perhaps some sequences could contort atoms in a way that would more quickly link the semipoles in a string. This atomic/plasma structure would in turn act as an enzyme. From there, perhaps either the beaded string or plasma structures would produce a structure that encapsulates both, i.e., something akin to a cell membrane. Thus, we'd have the three requirements for life: replication, metabolism, and compartmentalization inside the seemingly inhospitable environment of a star. So, that's the alternate biochemistry iceberg in a nutshell. It's very thought-provoking to envision how the fundamental chemistry of life could differ from ours in organisms who came into being on another world. I tend to think that since the rules of chemistry are universal, the biochemistry of aliens would be recognizable to us, but would have some subtle differences, like using some different amino acids in carbohydrates. This is especially true when a biotic process has an abiotic analog. At the elemental level, I think life will be based around the same major elements, but there are some situations where elements, which exist as trace constituents in our biochemistry, could play a role on par with some of the major ones. As for beings composed of energy within stars, well, it's tricky to disprove because it's based on largely theoretical physics. But while Carl Sagan described himself as a carbon chauvinist, I'm a bit of a matter chauvinist. Thanks for watching, and as always, if you enjoyed what you saw, please like and subscribe.